DNA replication. DNA replication begins at the origin of replication on both of the parental strands of DNA, which are separated to form replication bubbles. This separation is done by helicase hooking onto the strand of DNA being copied at the origin. This enzyme untwists the double helix, separating the two old strands. Along with helicase, molecules of single-stranded binding proteins line up along the unpaired bases, holding them apart while they serve as templates for the synthesis of new complementary strands. Also, primase begins to join RNA nucleotides to the old strand of DNA being copied to make the primer, which is about 10 nucleotides long. After primase is done making the primer, DNA polymerase catalyzes the elongation of new DNA, continuing on from the primer following the helicase towards the 5' end of the old DNA strand, which will become the 3' end of the new DNA strand. The main problem with DNA polymerase is that it can only copy in a 5' to 3' direction, since the DNA strands are anti-parallel, which creates a leading and a lagging strand. To compensate for the leading strand, which just continues to copy, the lagging strand is fragmented into several Okazaki fragments, which are primed by primase, elongated and copied by DNA polymerase, and joined together by DNA ligase. DNA polymerase will later go back and replace the RNA primer with DNA on both the leading and lagging strands. DNA replication is semi-conservative since the two strands of the parental DNA separate and each function as a template for synthesis of new complementary strands.